All right, so tonight we're being joined by Dr. Deborah Mash, who is one of the world's foremost experts on Ibogaine. She's the CEO and founder of, I apologize if I say this incorrectly, Demorex Incorporated, a clinical stage drug development company advancing Ibogaine and its active metabolite, Noribogaine, for the treatment of opioid use disorder. She's an NIH-funded neuroscientist, lecturer, and inventor who has contributed to studies of the brain and addiction neurobiology. She received her PhD in neuropharmacology from the University of Miami School of Medicine and completed a fellowship in neurology and neuroanatomy at Beth Israel Hospital at Harvard Medical School. And she is currently Professor Emeritus of Neurology and Molecular and Cellular Pharmacology at the University of Miami School of Medicine. I realized that I neglected to introduce myself. My name is Jordan Sloshower, one of the psychiatry fellows here at Yale and co-founders of the Yale Psychedelic Science Group. So really a pleasure to welcome Dr. Mash and turn it over to you now, Deborah.
Well, greetings from Miami, and thank you for joining this Zoom meeting to listen to a story that I'm going to tell you, which has really been a very long journey for me. And I'm always excited to share it with colleagues and people that are interested in Ibogaine and addiction. We've already heard my introduction, but by way of conflict of interest, I just want to mention that I do hold shares in the company which I am the CEO and founder of. And our company is working on both Ibogaine and the active metabolite of Ibogaine, Noribogaine. So a little bit about the history, just with this introduction slide, and then I'm going to take you on a journey with me down memory lane and then bring us to the present and where we're going with the company. Ibogaine has a very long history. The history goes back almost over 100 years, but it was actually marketed, many people don't know this, but it was marketed in France under the trade name Lamborine until 1970. The first discovery that Ibogaine could be a potential addiction interrupter was described by Howard Lotsof in the 1970s. And I first heard about it in the early 90s. And our group at the University of Miami went to the National Institute on Drug Abuse and then went to the FDA. And we actually received our first approval to conduct studies in humans. So first in human studies in a pharmacokinetics and safety protocol, but unfortunately we didn't advance due to a lack of funding. And I'll talk a little bit about that when I go through some of the data slides. About 10,000 people from our review have done Ibogaine, have taken Ibogaine in various settings. And this again, going back all the way to the time of Howard Lotsof. But they often do it in medical settings that are underground, kind of in the gray zone, and often not legally approved. Also, there often are not skilled practitioners. And this has led, unfortunately, to about 33 reported deaths in the literature. And we've had the opportunity at Demorex, working on our clinical trial application, to review those. So we maybe will get into a discussion of what that represents in the context of the discussion. In 2017, at the height of America's heroin and prescription drug epidemic, as deaths were increasing throughout the nation, I had to ask myself a fundamental question, and that was, I let Ibogaine stop, and perhaps it was time for us to finish what I had started. And because of that decision, I left my academic position at the University of Miami. I had been on the faculty there for over 30 years. I'm the founder and former director of the University of Miami Brain Endowment Bank an NIH-funded national neurobiobank, where we had one of the largest collections of postmortem brains in the world. And we had been supporting academics throughout industry and also academic investigators, NIH-funded investigators who were interested in studies, not only neurodegenerative, but also neuropsychiatric disorders. So I had a real connection with medical examiners, not only in South Florida, but throughout the United States. So I was well aware of the numbers of opioid intoxication deaths. And of course, we know some of this uptick was due to the fentanyl analogs that were coming into the United States. And of course, fentanyl, which is very addicting and very hard to break that habit, is currently a cut in with the heroin. So many young people were dying, many people who were just, getting into sort of the transition between prescription opioids to heroin. The numbers were staggering. And then of course we had opioid exposed infants that were appearing in the hospital. And I had to get back into the game, because seeing is believing, and I'm going to show you some of that data. If we look at the problem that we're faced in terms of the numbers today, it's estimated that about 40.5 million people in 2017 were dependent on opioids. And medications available for the treatment of opioid use disorder are methadone and buprenorphine, the substitution therapies, and the opioid antagonist, naltrexone. And this is what we have. Buprenorphine, when I started Demorex back in 2010, and at the time we started Demorex, and at the end of 2010, we were going to advance the active metabolite. We weren't going to take Ibogaine forward. And we could talk about that in the discussion section. But the buprenorphine sales in late 2010, early 2011 were about $300 million. Today, it's estimated in the last 2 years, if you go on the internet, it's about $1.7 to $2.3 billion. And the market share now by 2026 is estimated to be $4.5 billion. So when I first went to the FDA, and we spoke also with NIDA, pharmaceutical companies, Big Pharma was not interested in advancing medications for the treatment of addiction. Besides the problems with working with these patients and their comorbidity, which everyone on this call understands, there wasn't a market. And so pharma just wasn't interested. And believe me, I tried. I tried to speak to Big Pharma and went up in front of many groups and presented, but there was just no money to fund. We know today that the cost to, besides the loss of life and the disruption of the family and the way addiction affects the family, because it really is a family disease, the cost of the current opioid epidemic is increasing, and it's well over $1 trillion. And if you look at the aspect, even incarcerated people who are drug dependent, this is a big expense. So non-opioid treatment, some new medications, some alternatives to break the cycle of opioid dependence are needed. This is one of my favorite slides done by Christopher Evans from one of his publications. And I think it really helps us to understand not only the importance of medications for the treatment of withdrawal, but also the neurobiological underpinnings. So we know that when people who abuse opioids, short-acting opioids especially, go through these kind of mini kindling cycles of getting high, getting the euphoria, and then going through the wearing off cycle and into the first stages of withdrawal. And of course, with that comes the anxiety and the dysphoria. So it's these up and down cycles of getting high and wearing off. When you try to go through withdrawal, and keep in mind that many people do have a very difficult time successfully detoxifying from opioids, they're at risk. And so that next time the life stress comes in, they have this kindling again of the loop, and they're back out and they're back using. And when they feel bad, whether it's the post-acute withdrawal symptoms or perhaps the craving, which then triggers the memory of the withdrawal syndrome. So we know, all of those of us that are the neuroscientists on this call understand that there are many different cognitive domains that contribute to this, and the neurobiological underpinnings involve multiple areas of the brain. But those triggers to relapse have a lot to do with the memory, not only of getting high, but the memory of the pain of withdrawal. What's available today, what clinicians use for withdrawal are methadone taper, buprenorphine taper, which is, even in maintenance therapy, a tapering off of buprenorphine is always a goal for many patients. And then, of course, off-label is clonidine, the alpha-2 adrenergic agonist. In 2018, the FDA approved lofexidine as the first non-opioid treatment for management of opioid withdrawal symptoms in adults. I won't share those data with you today, except to tell you that obviously the roadmap for lofexidine approval in the United States, the efficacy endpoints are extremely important for us and are guiding the Ibogaine protocols that we're going to be going up in front of the regulatory agencies with. But if you look at the data, they're not stunning. Yes, they show improvement, but many patients, again, return to drug use, and they have a difficulty completing the actual detoxification. As part of our review and preparation for submission, we had an independent audit review, and I went back deep into our clinical records. And in 2018, again, finishing what I started, I published this paper with my colleagues from the University of Miami and St. Kitts, which reported on 191 subjects. And this publication today is one of the largest observational case series in patient volunteers, and the data I will show you. And this was done on the island of St. Kitts with government approval, and most of this data actually has been submitted already to the FDA. So Ibogaine, I mentioned, is an old molecule. It is an indole alkaloid, and if you look at that O-methyl group on the slide, you can see that part of the molecule looks like serotonin. The other part actually has some opioid moiety to it, and there also are cholinergic parts of the molecule. So it right away, when you look at this as a chemist, you get the feeling, without doing in silico modeling, that you're going to be hitting this constellation of targets. But it's kind of interesting. This comes from Mother Nature. It comes from the roots of Tabernanthe iboga, which is found in the western equatorial parts of Africa and the region of Gabon and Cameroon. And it is used in high doses in Africa as part of the Bwiti religion, which is an Afro-Christian syncretic religion, and it is their active sacrament. This is actually a photograph of a woman who's preparing for an iboga ceremony. And I had been, when we started getting publicity originally back in the early 90s, I was contacted by some of the Bwiti bishops who invited us to come and visit. I never went to Africa, but I did have some other people who went and actually got to see the ceremony. So this is a little bit more of an expanded version of Ibogaine, and I mentioned about Howard Lutzoff, who made the seminal discovery, himself being an addict, and he took a dose, and he was a heroin addict who took a dose of Ibogaine and made the seminal discovery. He repeated that experiment with several of his friends, and they too had this withdrawal relief, and many of them reported that they actually stopped taking their drug of choice, and they were able to maintain sobriety for months to years. In 1985, Lutzoff comes back and decides that he's going to start a business, and he gets the first US patent for Ibogaine for opioid dependence. And there were 4 more patents that followed, one for nicotine, alcohol, psychostimulants, and polydrug dependence. He started an underground railroad of addicts helping addicts, and they went to Amsterdam. And there they were doing Ibogaine treatments in collaboration with a psychiatrist by the name of Dr. Bastians. He also went with ICASH and the group, and keep in mind at the time, HIV/AIDS activists were also going before NIH and before the FDA, and they went up in front of the NIH and they put some pressure on to get NIDA to start to think about funding. At the same time, in 1991-92, I submitted my first NIH grant to NIDA, and we were funded. And at that time, we were working on the cocaine epidemic. Miami, because of our close proximity to Central and South America and the transshipment of cocaine through the Bahamian corridor, we were the front end loading of the cocaine epidemic. And again, I was running my brain bank and working with the medical examiner's department, and we got interested in trying to look at CNS mechanisms of cocaine-related sudden death. While we were doing that, we got some national publicity and press, and I was contacted by Howard Lutzoff, so we were actually able to, with an invitation from him, to go to Amsterdam to see Ibogaine administration in person. And because I saw 3 young men, detox off of their drugs of choice, 2 of them, one was on 100 milligrams of methadone, and the other young man was injecting a large amount of heroin. He was a musician, a very talented young man. And the 3rd young man was freebasing cocaine. We were there and got to see the treatment, and I couldn't believe it. And we looked at each other and we said, how is it possible that 1 dose of Ibogaine can block the signs and symptoms of withdrawal? And not only that, to have this prolonged effect on these people such that they actually had insight into their addictive behaviors, and they were ready to start to think about changing their life, formulating a new plan to get off of drugs and stay. I'm incredibly eager and went back to my university and spoke to my university colleagues and said, you know, we need to submit something to the FDA, and I need to go speak to NIDA. So we did. And in 1993, we went up in front of the Drug Abuse Advisory Committee and got the first permission to go forward. The reason that they restricted us to Ibogaine veterans was because data that had come out of in rats that Ibogaine may cause cerebellar toxicity. And it was Mark Molliver and his group there who were NIDA contracted investigators who had been working, by the way, on MDMA toxicity, who then said, well, it's causing this cerebellar Purkinje cell dropout in animals. Nonetheless, we made it past that hurdle, and in 1995 I came back to the FDA and we got the full green light to go forward under an external DSMB. But unfortunately, the clinical trial was delayed because I was unable to fundraise it. I couldn't get a NIDA grant approved, despite the fact that I had worked with NIDA on the protocol. And also I was unable, Howard Lutzoff had the patents, so he would have needed to fund this from the private sector, and he was unable to do that. He was also difficult to work with, and I don't believe that there was ever a CRADA signed with the National Institute of Drug Abuse, but you know, it just didn't happen. It didn't go forward. So we were unable to proceed. And this was a great setback for us. So what I decided to do was to do something a little bit bold and unheard of at the time, but I did it with permission from the University of Miami, and that was that I went offshore. And we raised money, we got government approval, and I worked with a wonderful biological psychiatrist, Dr. Frank Irvin, who had been on the faculty at Harvard and was then on the faculty at McGill University. And he had a primate study colony down in St. Kitts, and I had worked with him in the past. So I asked him if he would help me and if we could start to do some observational open label clinical trials with informed consent in patient volunteers who elected to come down to St. Kitts and also to give us consent to collect data to submit safety, pharmacokinetics, and open label efficacy data to the FDA and to share with our colleagues. And so we began. One of the first things that we did was to demonstrate that Ibogaine was converted to a metabolite, and you can see the metabolite noribogaine is O-demethylated on the 12 position. And it is actually a large concentration of noribogaine in the blood. And as you can see in this slide, this is the classical pharmacokinetic profile for Ibogaine, and this was taken from a male patient volunteer. The Ibogaine is shown in red and the noribogaine is in green. At about 10 nanograms per mil is the cutoff of the machine for the level of detection. So what you can immediately see is that in about 15 or so hours, you're clearing most of the Ibogaine from the blood, but the noribogaine is elevated, and markedly so, with a long terminal half-life. But Ibogaine also has complex pharmacokinetics, and we figured out early on that it was cytochrome P450 2D6 metabolized. So again, the first panel shows the fast metabolizer. The second panel, SKM1, is a poor metabolizer. The third panel below, which is the female, SKF1, is a heterozygote. And then SKF2 is an ultra-rapid metabolizer. Now, these data were really amazing for us because our first subject who took Ibogaine in St. Kitts was a poor metabolizer. And this is really like the luck of the draw because there are only about 3% of the general population have the cytochrome P450 2D6 deficient genotype. So the very first person we give Ibogaine to has long effect of Ibogaine. So he had the oniric effects of Ibogaine for a very long time, all the way through the night, and he was ataxic right up until 24 hours. The ultra-rapid metabolizer, the person, the female at the bottom, had no visions, but she had full blockade of her heroin withdrawals. So we knew immediately in the first 6 patients that we ran in St. Kitts that the Ibogaine was responsible for the visionary phase of the drug. And it looked like maybe some of the purported efficacy of the treatment may be due to the activities of this metabolite. And we did in vitro work with Dr. Scott Obach from Pfizer in a collaboration, and he ran an incredible laboratory there at Pfizer, one of your neighbors close, the Pfizer facility close to you, in fact. And he demonstrated that, of course, most of the metabolism is first passed through 2D6, and there's about 10% going through the lesser cytochromes. And you can see again here when we look at this population plot that we have the 4-4, which is the poor, the intermediate, and the fast metabolizers. So complex pharmacokinetics. At the University of Miami, we were able to do a few of our patient volunteers under our FDA-approved study. And this was the 2 milligram per kilogram data, again, which shows on the bottom that noribogaine has a long half-life, as shown in green there. It's anywhere from 17 to 20 to 24 hours. And the mean residence time in the body is much longer than the Ibogaine, as is the area under the curve. So that was a clue for us because we thought, well, if the Ibogaine is being cleared, then maybe this time course, especially when you're looking at people who were maintained on methadone and have a longer breakout of withdrawal symptoms over time than short-acting opiates, that maybe some of the efficacy of the Ibogaine treatment was this kind of 1-2 punch between Ibogaine and then this long-lasting tapering effect of the active metabolite. And this makes sense too, because it's not only the acute signs and symptoms, the cold turkey symptoms that we show here of the muscle aches, the anxiety, all of the adrenergic symptoms, the yawning, the tearing, the sweating, advancing up to the nausea and the diarrhea and the intense muscle cramping, but it's also the post-acute symptoms of the depression, the drug craving, and the anxiety. And so even if you can get past the first phase of the withdrawal, you then have the horrible dysphoria, the anhedonia that you feel in those days up to the week after you try to detox off of the opioids. So we saw the Ibogaine block withdrawal symptoms, but again, we did this with our physician ratings of using the objective opiate withdrawal scale in humans. And we could demonstrate that if you looked at the pre-Ibogaine AUS scores, that you could just really knock these down. And the next slide that I'm going to show you, and I'm not going into detail, is included in the publication. And these are representative data showing males and females and their pre-dose and post-dose scores. And you can see that we have a range of scores here based on where they were in terms of beginning to break withdrawals. But the thing that was just amazing is that people did not have withdrawals. And not only did they not have withdrawals, but you know that when you do detoxify patients and they're early in treatment, one of the main problems is that they split. They don't complete the detoxification, they split. And many of them who get into early rehabilitation also split because they're still going through the withdrawal symptoms. We did not have people jump the fence in St. Kitts and go downtown to score drugs. And we kept waiting for that to happen, needless to say. And we had 2 psychiatrists who were part of our group. And we had therapists, and we had case managers, and we had counselors. The people that did the detox stayed in treatment and completed the detox. One of the other things that was really remarkable for us, and again in St. Kitts we were we were living with the patients, so we were eating meals and whatnot with them, and they would come to us and they said, Dr. Mash, 2 to 3 days out, what's in me today? I feel really great. You feel really great. Okay. Well, part of that was the depression scores, and of course comorbidity. All of you on this call know that the comorbidity with depression and generalized anxiety disorder in opioid dependent patients is very high. And in fact, in our group, SCID ratings with the psychiatrists demonstrated that about 50% of our patients who were opioid dependent would meet criteria for depression. So we were able to demonstrate that the scores went down in opioid-dependent patients, and although the ratings were lower in the cocaine cohort, we also saw the same trend with the cocaine-dependent patients. Craving, we know, is definitely linked to mood. So it's not only the fear of the withdrawals that will trigger the craving, but also mood scores and suffering from the depression, the learned helplessness, the anxiety. In addition to the withdrawal fear and the learned withdrawal is also what's putting people at risk for relapse to craving. So we thought this, again, would be a benefit of the Ibogaine, a benefit above and beyond a standard opioid taper or a clonidine or lofexidine taper, because we were getting this benefit in terms of improvement of mood scores. We use the heroin craving questionnaire now, assessment that was developed by our colleagues at Johns Hopkins. And we looked at our patients, and the data was broken out in terms of four factors that we were very interested in. One, again, being negative mood state. Second, the desire and intent to use the drug now. Lack of confidence in the ability to quit, and expecting positive benefits. And we were able to demonstrate that during the course of treatment on island, this decrease in craving measures. For those patients that we were able to follow up, who were able to be seen for assessments in Miami, and you see the follow-up numbers that we had at 1 month, craving scores were low. So this again suggested that the Ibogaine treatment not only was affecting mood, but was also having a persisting effect on craving for drugs. And of course, the patients also gave us verbal endorsement of this. I didn't need to get high. I didn't want to get high. I don't feel like getting high. Again, the data here shown for the cocaine-dependent patient volunteers, and we used not only the Hopkins cocaine questionnaire, but we also used the Minnesota cocaine craving scales. And you see there on the bottom, again, with both assessments, a decrease in cocaine craving. And cocaine craving is, the main thing that seems to bring these patients back out, because they don't go through the withdrawal severity that you see with opioid dependence, albeit there is a withdrawal syndrome with cocaine as well. So what is the mechanism? What would explain this? You give a single dose of Ibogaine to a patient. They have visions anywhere from 4 to 8 hours, depending on their metabolizer status. They then go into a deep phase of cognitive introspection. They can stay under medical monitor for 24 hours. The next day they're up, they rest a little bit, and by the afternoon of the first day out of the clinic, they're willing to go in and sit down and talk to their counselor. Dr. Kleber, when I first told him about this before I went to the FDA, he said, "Deborah, you're describing Paul on the road to Damascus. No one will ever believe you. Don't work on this. Do not study Ibogaine." Dr. Kleber passed away a year or so ago, but he stays in my mind, and I think of him often, because he was so adamant that my career would end if I took on Ibogaine. But as a neuropharmacologist, I was really intrigued by this. I wanted to know what was the mechanism of action. And if Ibogaine was a lead molecule, maybe we could synthesize some other molecules. We could have a whole group of molecules, and we would learn something about the brain and the neurochemical underpinnings of this, and it would be great. So I wrote some more grants to NIDA on the idea of medication development with new classes of Ibogaine-related congeners, much like Stan Glick was doing with the 18-MC, 18-methoxycoronardine. We'll talk about that during the discussion, no doubt. But I couldn't get any traction from NIDA on that either. So we had to do a little bit of our own work, and we did. And I'm not going to go into all of this with you, but and I'll try to summarize it because it's too much. But the interesting thing about noribogaine is that noribogaine is a biased kappa agonist. So, there was a lot of interest at NIDA on kappa drugs, but kappa agonists are what? Every psychiatrist on the line knows, they're dysphoric, right? It's kind of the yin-yang. Mu is to euphoria like kappa is to dysphoria. But those kappa receptors sit right on the dopamine neurons. In fact, they sit on the cell body fields of all monoamines, serotonin and dopamine. So they regulate the kind of set point. And there were some fundamental, beautiful papers that were done on this that show that it's also part of the gating mechanism between the crosstalk of dopamine and the opioids in the striatum as well as other parts of the brain. So this was a very important paper, and Emily Millay was the lead author on this, and did an amazing amount of work for us, along with some of my other people who worked and people from the University of Miami. But this paper was really a lot of fun for us because it said we've got a partial kappa agonist. And not only that, it's not recruiting beta-arrestin, which is very important for for opioid tolerance. So the crosstalk between mu and kappa and this unusual pharmacology of Ibogaine gave us some clues. Now, Stan Glick adds a really important piece of the puzzle when he says that his molecule 18MC acts on alpha-3, alpha-5 beta-4, alpha-3 beta-4 receptors. And these are very important because they sit in the habenula, and the habenula puts the brakes on dopamine-mediated reinforcement. So there's a lot of interest in NIDA and some very interesting animal studies who have looked at this mechanism, but if there hasn't been a lot of drug development around molecules. And in fact, at Demorex, early on, we ran our Demorex 1001A and B, which are two forms of the Ibogaine. They're ansolvates. We hold intellectual property around this, the preferred ansolvate. But you see that the green on the bottom panel overlaps with mecamylamine, which is the prototype antagonist for this, which is a pharmaceutical. But the 18MC, which is the obiter molecule, is shifted to the right, meaning that it has a very low affinity. So Ibogaine even has higher affinity than the prototype developed by Stan Glick. So another clue. So what we think is that you've got a mix of Ibogaine and Ibogaine. It's a pleiotropic action. Ibogaine acts on NMDA. So you get the NMDA reset, which probably helps with the depression. As we know, esketamine, Romalda's molecule, the dextromethadone, the renewed interest in NMDA drugs for intractable depression. Atai has R-ketamine. There's a lot of interest in this space. Ibogaine acts on NMDA, and it also acts on serotonin receptors. The Ibogaine acts on the serotonin transporter, elevating serotonin in the synapse, and it has this interesting effect, atypical opioid pharmacology, and then finally acting on the nicotinic receptors in the habenula. So it's these multi-target actions of Ibogaine and Ibogaine that we think may explain how only a single dose of Ibogaine in humans can block opioid withdrawal, improve mood, and alleviate withdrawal-related dysphoria, and that this persistent effect is probably due to the slow washout of the metabolite over time. And we have other data from the Ibogaine clinical trials, which shows that indeed Ibogaine has a long half-life. So this, again, gets me very excited because, you know, when I started, no one wanted dirty drugs, right? They wanted drugs that would act on a single target in the brain. But in fact, most of the good CNS drugs are ones that hit multiple targets. And addiction as an acquired disease involves a circuit level reset. It's not just dopamine transporter. That was the mantra at NIDA for many years when I was beginning my research as a NIDA-funded investigator. We all wanted a dopamine-sparing cocaine antagonist. Forget about it. It's not going to work. So you have to have a mechanism where you can modulate more than one target to reset this loop. And for me, the effect on mood and anxiety, that this treatment addresses that comorbidity, and that it has other effects that implicate other drugs that are abused, sounds to me, at least, as a pharmacologist, like something that could be an advance. Because you're not only talking about mood, you're talking about reward, you're talking about resetting opioid tolerance. You're able to block withdrawal, you're able to block the cravings. And the question is, will it show an efficacy in terms of relapse? And that's a fundamental question. do patients who successfully detox, when you start to run large clinical trials, what's going to be their rate of return to drug abuse? Some animal studies. So Ibogaine, this is like reverse translation. So we were able to publish a paper a few years back where we demonstrated that Ibogaine shows high brain uptake and anti-withdrawal effects in rodents. And this is the classic naloxone-precipitated withdrawal. I was not blinded to condition, but the raters in our group were, so they didn't know whether it was placebo or active. We did a dose run-up of 1030 up to 100 mg per kg in these mice. It was naloxone-precipitated, and you can see dose-related decrease when you give Ibogaine to these mice. So proof of concept. One of the questions, of course, that you have to address, that we're going to have to address with Ibogaine, is abuse liability. The same thing for Ibogaine, but Ibogaine has no abuse liability. We'll come back to that. But Ibogaine, we didn't know, and we didn't have the self-administration studies with Ibogaine. So we did the CPP model, the condition place preference, and what this slide shows is that there's no difference up as you escalate the dose of Ibogaine compared to vehicle. So it doesn't show preference. So this suggests that Ibogaine is not going to have abuse liability. It's not going to be addicting. Sue Shank, my very dear friend and colleague, she ran the NIDA, one of the NIDA screening programs for the Medication Development Division. I asked her years ago, I said, Susan, take Ibogaine and run it through the rats and see what happens. And she did, and she demonstrates here that if you give Ibogaine to rats who are self-administering at very high rates cocaine, they'll stop taking the cocaine, and that the rate of self-administration is even decreased at 24 hours after you give the drug. But she also shows here on the right that it doesn't affect sucrose. These are data from Amir Rezvani, and they're some of my very favorite data. Dr. Rezvani has been an NIAAA funded investigator, and he had published a seminal paper on Ibogaine, along with the colleagues out in California at UCSF, Dorit Ron and her group at the Gallo, and they both groups demonstrated that Ibogaine would rats that were self-administering alcohol would stop taking alcohol. So these are those high alcohol preferring rats, good animal model. I asked Amir if he would run Ibogaine in the same assay, and he did. And here we show dose-dependent decreases in alcohol self-administration. Dorit Ron published a paper several years back where she also looked at Ibogaine and demonstrated that Ibogaine, but not 18-methoxycoronardine, will block alcohol taking behavior in rats. So again, here's another suggestion that these molecules are doing something that's generalizable to addiction. It's acting in the addiction circuit. And last but not least, we were able to, again, with my former postdoc and colleague, Dr. Mallet, demonstrating that Ibogaine will stop rats from taking nicotine, and it looks pretty good compared to Chantix. There's something to this. So we have a new chemical entity with Ibogaine. We got intellectual property around the molecule. It targets drug sites, as I've shown you, multiple drug sites in the brain. It has effects in animal models of drug and alcohol taking. What are we going to do with it? Originally, we were going to develop it for withdrawal-based therapy and perhaps office-based therapy. But we're rethinking it now, and you'll see when I come to the end of the slides why. But right now, we're thinking about it as possible an adjunctive therapy post Ibogaine detoxification. And we're revisiting the target product profile and our clinical development plan. So to summarize, both of these molecules seemed to play a role and seemed to be important for development. We would argue that there is an unmet need for effective non-opioid treatments for patients. Morphine are extremely important for patients. It saves lives, we know this, but substitution therapy is often diverted. Chronic use of methadone leads to cognitive issues. So is there a possibility? Is Ibogaine a possible transition to abstinence-based therapy? And could we add on Noribogaine to maintain patients during early recovery? So we would argue yes, that medically managed withdrawal, we know, is often a necessary first step in recovery for patients physically dependent on opioids. And we would also argue that Ibogaine, perhaps because of its oniric effects, will also give us better outcomes, not only in terms of management of withdrawal, but in rates of recovery for patients. In other words, successful detoxification as they make a transition to abstinence. To try to get some understanding of this when we were in St. Kitts, we used what was called an elicitation narrative. And the elicitation narrative was developed from anthropological techniques. I had a colleague who's chairman of the anthropology department at the University of Miami. He's one of the co-authors on the publication. And so what we did is we talked to them and we videotaped them. We gave them a structured set of questions, and then they would answer us, and then we had the people come in and do the content coding. We had 2 people code the content. When they got up to 90% agreement, we ran the analysis. And the common, things that the patients reported to us is that, yes, they thought Ibogaine was useful for their drug problem. They felt they had been given insight. They believed they needed to get sober and abstinent. They felt cleansed, rebirth. So they had this kind of aha oniric experience from the Ibogaine. It was very powerful for many of these patients. They felt they had been given a chance. And keep in mind, the people who came to St. Kitts, these were chronic relapsers. These were not, people who just started using drugs. These were people that had 10, 12 years of hardcore drug use, who had gone into treatment again and again, some 4, 5 times, had ODs. I mean, on the ASIs, on the Addiction Severity Index, they ranked high. The other thing that was really interesting here was they didn't really like it. You're saying, want to take Ibogaine again? No, it's not MDMA, it's not psilocybin, it's dark, and it's a little scary. So we can talk about that, but it's not something that people don't abuse Ibogaine. Nobody's going to a rave scene and taking Ibogaine. Anyway, you couldn't walk if you took Ibogaine. You'd have to lay down. So in the end of this year, Demorex, the company I founded, took over and came back in and took over after I had left it for a number of years, signed a joint venture with Atai Life Science. And many of you on this call know who they are, but their interest is in Ibogaine and working with us. And they're a wonderful group of collaborators, and we're really enjoying this opportunity, and they're very important to advance Ibogaine to the clinic. But their question is, what they would like to demonstrate is whether or not Ibogaine detoxification is a transformative therapy for opioid use disorder. And I think we may prove that it is. As I talked about the pharmacology, Ibogaine and Noribogaine reset. It's a neurobiological reset. Think about brexanolone for postpartum depression. It's almost like Ibogaine is to glutamate reset, like brexanolone is to GABA. It's resetting in different cognitive and emotional domains, and that this reset is part of the efficacy of this treatment. Motivation, patients show motivation for change. Intractable people who said, I'm not going to meetings. We are not going to meetings. I don't want to, no, it doesn't work. Became motivated. They started to think about. They had an ability to revisit where their addiction took them and to start to think about what they could do to not fall back in the hole. And this has got to be part of it, right? So when you think about the psychology of this, it's addiction is a biopsychosocial disorder, and you need to have the behavioral component. So it's very likely that Ibogaine may prove to be effective as an intervention for opioid use disorder, precisely because the oneric effects help to prepare patients for behavioral support and post-detoxification treatment. They get back their control. They're able to engage the frontal lobe. And with a good therapist and a good modality to match to the Ibogaine, this may prove to be a transformative molecule. We are submitting our CTA, and with the idea again that Ibogaine hydrochloride can become a treatment for opioid dependence, because not only of its pharmacologic, but also because of its effects on behavior as a behavioral intervention. We believe we're going to show harm reduction, and that will improve the quality of life and well-being for patients who want to break the intractable cycle of use. The CTA will be for the treatment of acute opioid withdrawal symptoms and facilitation of abrupt discontinuation of opioid use. And if that guy on the left doesn't stop me, if COVID-19 doesn't stop us, we look to have our first patient enrolled at the end of 2020. I'm really always delighted to have the opportunity to share my journey with you. It has been a long journey for me, but I'm really excited. Again, I was excited at the beginning, and I'm perhaps more excited now because of the new renaissance and interest in the psychedelics that are being used therapeutically with qualified physicians, that Ibogaine may have another shot on goal. And again, I want to acknowledge there are many wonderful people that have been with me on this journey that have helped me for decades, actually, and I can't name all of them. I've lost a few of them. They're great colleagues and collaborators, and I think very fondly of them, and also to acknowledge Demorex and my colleagues at Demorex, and then also the group who has joined me from Atai Life Science, specifically my Chief Medical Officer, Dr. Matthias Luz, Gregory Bates, who's our lead regulatory advisor, and then Dr. Srini Rao, who is the CSO at Atai. Thank you.
Thank you so much, Dr. Mash. That was a wonderful presentation. I have so many questions to ask, but I'm going to open it up to our participants here. And so just a reminder, everyone, if you have questions, either post them in the chat or preferably click on your participants button at the bottom of the screen and then use the raise hand. There should be a button that says raise hand. If you raise your hand, I should see that and I will unmute you and you can ask your questions. So while folks are doing that, I guess I'll start it off. few questions I would love to ask. I guess one burning question is, how, and I'm sure this is a question that you've already faced, is what you're proposing with Ibogaine is a abstinence-based paradigm. And of course, the current sort of standard of care is the replacement therapies. And with fentanyl being the preferred drug on the market, obviously some concern, and you mentioned this, that if patients do return to drug use after detox, that there could be that risk of overdose there. How do you think about that issue?
We think about that a lot. I mean, obviously the standard of care now is to put patients quickly, as soon as possible, on substitution therapy. And in many parts of the United States, patients, especially in the rural parts, they can't get methadone. And buprenorphine providers are not available. Good luck trying to find an addiction psychiatrist. And then you've got incarcerated patients. You have people who are going through withdrawals with, hardly palliative therapies. Give them an Advil and let them get sick and give them a bucket. So from a humanistic standpoint, I think that the molecules have a place. But patients should have the opportunity to transition to abstinence. Naltrexone, which I happen to be a big fan of, and many patients can't make it through the detox to get on the naltrexone. There's no issue with transitioning Ibogaine patients to naltrexone, I can tell you this. And perhaps the noribogaine in a low-dose formulation will be the add-on molecule. So they won't be flying without a safety net. But we had patients come to us, some chemically dependent patients who had gone up on their tolerance to opioids, legitimate pain patients who then started, taking too many opioids, who wanted not to go on buprenorphine, but wanted to get off, and they couldn't get off, and their doctors tried to detox them. I mean, and these were competent clinicians who knew how to do it, and they couldn't make it. And then they go and they do the Ibogaine, and they make it. Mm-hmm. So, and I think this will be a clinical judgment. The doctors should have the tools to give their patients choice. Together, you make that choice with your doctor, with your clinician.
Awesome, thank you. We have a question from Jeremy. Jeremy, do you want to ask your question?
Yes, thank you, Dr. Mash. That was an amazing and wonderful and powerful presentation. It was truly excellent. You hinted that you were going to be asked a question on 18MC, so I'm going to ask you the question about your views on the efficacy of 18MC, bearing in mind what MindMed are planning to do. And then secondary question quickly, your views on FDA's perspective on the HERG channel blockade issue with Ibogaine and noribogaine.
Yes, these are very fundamental questions. So two great questions. On the 18MC, first of all, from the bottom of my heart, I wish them luck. I hope that they are successful. The 18MC data in the animal models do not show me the same signal the robust signal that was described by Stan Glick and many others with Ibogaine in the rodent models. So from a animal test tube perspective, I do not see the affinity at the target. I showed you that data. I haven't published that data. I don't see the affinity at the target, and I don't see the stunning efficacy that Stan Glick showed in his early papers with Ibogaine. Now, having said that, And if you look at the way the publications evolved over time from that laboratory too, they started going towards obesity as an indication. So I don't understand that. Also, NIDA gave Savant $6 million non-dilutive funding back how many years ago to put together the clinical trial package, get the GMP product ready to go for first in human studies. Where are the data? Where's the study? It never happened. So to me, there's something missing. I wouldn't bet on it working for opioids. Will it work for other drugs of abuse? It may. But I right now, with the limited information that we have, and there may be other data out there that I don't see, but from the published literature and from the work that I've seen over, since Stan Glick's first reported on the molecule, and studies that we've done, some parallel studies in the Demorex laboratories, I'm not as optimistic for them, and I hope I'm wrong. And I say that sincerely. It's not a competition. People are dying, and we need medicines for addiction. I hope they succeed. The second question was the hERG channel. The hERG signal is a big stumbling point, no doubt about it. The FDA is well aware of protecting human subjects. We did not in St. Kitts we did not have a death in St. Kitts. Thank God. Thank God for that. My colleague who I trained in, who started when we closed St. Kitts because we wanted to come back into the United States and work in our country, I trained a doctor a Mexican physician, an emergency room doctor who's also a member of ASAM, and he started a detox program in Mexico. He has done over 1,500 to maybe 1,800 treatments. People give all these different numbers, but I know for a fact that he has done around 1,500 treatments. He has not had an Ibogaine death. So the QT prolongation, it's very important to, as part of any safety study and efficacy trial, to demonstrate to the regulators, to demonstrate to us that this is dose related. Most of the so-called Ibogaine related deaths, and obviously many of those deaths people had comorbid cardiovascular conditions which would predispose them to death. This is definitely a cardiovascularly active molecule. It has to be given under full medical monitor. It has to be given in a safe setting. But we believe that it is any adverse signaling through the hERG will be dose related. So we can be at a blood level that will not give us the QT prolongation signal that would put patients at risk. What I shared with you on the JT peak comes from the FDA. So I'm excited about that. I can't wait to get that data for us to learn whether or not we have a balanced drug that is not acting on multiple channels in the heart. And that's why we didn't see it. The other thing I want to mention is that it may be that it's very hard to disentangle Ibogaine from Noribogaine. We have some ideas about this, of course, but when you look at patients, you're looking at the combined effect of both molecules in the blood. So the effects on the heart could be due to Ibogaine or they could be due to Noribogaine. Well, it may be that the Noribogaine has more of a hERG prolonging effect than the Ibogaine itself. So we're going to sort that out. Does that answer you? Yes.
Thanks for that. I have a question from Mustafa here. He asked, in your opinion, is there any difference in effectiveness, or I would just add in effect, between the naturally sourced Ibogaine or from the Iboga plant versus the synthetic version?
Right. I would say absolutely no. All right. Keep in mind that when you take Ibogaine hydrochloride and I've had both the Ibogaine natural product from the roots, purified from the roots of Tabernanthe iboga, and we made it into the hydrochloride salt. That molecule is no different, it behaves no different than if you have production through a semi-synthetic route. So obviously no one is going to go dig up all the Ibogaine plants, because first of all, you can't. That's number one. Number two, it's in a rough region of Africa right now, where Boko Haram and other groups are running around, and the government is unstable, et cetera, et cetera. So you're not going to source Ibogaine from Gabon and Cameroon. It's just not happening. The semi-synthetic production of Ibogaine is a well-validated route to both Ibogaine and noribogaine as well. And so we have GMP production, we have full control manufacturing and chemistry mapped out that will be acceptable to the regulators. And in terms of the pharmacology and the experience of patients, we don't expect there to be any difference.
Thanks. And while we're talking about the molecules, I wanted to ask you about patents. You'd mentioned that you were pretty much discovered noribogaine and held the patent for that, and that Lotsoff held the initial patents for Ibogaine. So now, as you're moving forward with Demorex, can you just tell us how you're thinking about patents? Are there any patents for the molecules, for use patents? How do you think about intellectual property in this?
Yes, so intellectual property is obviously important for drug development, because if you don't have the money, you don't get into clinical trials, right? So you have to, you can have exclusivity. The first to market would have, 5 years exclusivity in the United States, 10 years in Europe, under Hatch-Waxman, 5 years. So it's not a lot of time. And most companies, every pharmaceutical company I spoke to wanted to know that you had the IP. Yes. So that's extremely important. Noribogaine, we have a robust portfolio around noribogaine, and we're working on new IP around Ibogaine. But Ibogaine will be much more difficult, obviously. So for us, it will be first in the clinic, and once we get in the clinic, then we anticipate that IP will flow from that. And we also, working together with the ATI collaborators, will be working on IP. It's a very important part of what we're doing. Yes.
And so those would be patents for the molecule as well as use patents, or mostly the use patents?
I'm not going to discuss on this call intellectual property plans for Ibogaine.
Gotcha. All right. Dr. Gary Rudnick has a question. I'm going to unmute you, Gary.
Hi, Gary.
Hi, Debbie. So my question is that about the pharmacokinetics. You showed a really striking difference between the time course of Ibogaine and noribogaine in human subjects.
Yes.
Can you say anything? It was hard for me to sort of get a picture from looking at the graphs, but can you say anything about the absolute efficacy or potency between the two in those studies? And the reason I ask is that I see this whole thing from a very different kind of point of view. My main interest is in serotonin transporter. Mm-hmm. At that target, at least, a noribogaine has a higher affinity.
Yes, yes.
And so I'm just wondering if that translates at all to the results you saw in the patients.
Well, you've published seminal, and you are Mr. Serotonin. It's good to see you. I haven't seen you in a while.
I know.
Yeah. I've been busy. It's 50. You've published your numbers. I have my numbers. I mean, yeah, Ibogaine is a serotonin reuptake inhibitor, and a kind of an interesting one. And had a paper with Randy Blakely recently as well. But on that target, which contributes probably to mood and some of the craving reset, I think, that it's probably being mediated through the Ibogaine. But remember, if you're looking at depression and that fast reset on depression, you've got Ibogaine on NMDA.
Well, yeah, I'm just wondering how much of that difference in affinity between Ibogaine and Ibogaine shows up as a difference in potency in your studies, if you can say anything.
In the
about
humans, we can't.
Okay, so
in the humans, you can't.
It's comparable, in other words, the potency.
You would have to run Ibogaine without the conversion to the Nor.
Yeah.
Yeah, we'd have to block it.
Right.
Right. And there's kind of a way to do that, and I've thought about it, but.
Yeah, okay. No, that's fair enough.
It's good to see you. Thank you for coming to listen.
Oh, I wouldn't have missed it.
Jordan?
Sorry, I'm muted. We have a question from Melissa Weimer. Melissa, do you want to ask your question?
Sure. My internet connection's sort of bad, so hopefully you can hear me. I was just curious. I've actually had patients try Ibogaine. They go to Mexico and they utilize the Ibogaine detox there. And some of them have had very unsuccessful courses. I actually haven't had a single patient be successful. So I was just hoping you could tell us a little bit more about the patients who didn't do well, what you think is mediating that.
So when you say, Melissa, if you would be kind, were they detoxed? And when did you see them afterwards?
They were detoxed, and I saw.
So they were detoxed successfully, but then they relapsed at some point. Is that what you're saying?
Yes, and quickly after. So I'd say, within 3 to 6 months.
Within 3 to 6 months, they detoxed. Well, they detoxed and then they relapsed 3 to 6 months. Yeah, I don't think Ibogaine's a cure. Okay, we have patients that are clean and sober since 1996. Is everyone clean and sober? No. Do patients relapse? Absolutely. Okay. The fact that they were able to get, and I mean, I don't know the comorbidity of your patients, how many patients you've seen, what their aftercare was, if they had other comorbidity with depression or bipolar disorder, if they needed to be back on medications, so it's not a magic bullet. It is not a cure. No. Can it be combined? We're developing it for detoxification. Does it need a behavioral component to it? Yes. Do some patients need more than one Ibogaine treatment? Maybe. Should they come back and take Ibogaine at 6 months or a year later? Could be. These are the questions we don't know, which is why we've got to get in the clinic and we've got to begin to understand more than the observational data. And the other thing is that the patients need to be treated with the same protocol. Because when you go to Mexico, you don't know how much of the drug they got. You don't know whether they were a good match for it to begin with. You don't know whether they're polydrug dependent. We don't understand their comorbidity. So, again, this is why pharmaceutical companies don't want to work with this cohort, because it's really hard to get a well-matched patient cohort to do a well-controlled efficacy study in humans. Looking at the return, I'll tell you, Melissa, if we looked at right now those numbers that you just said against lofexidine and and their Kaplan-Meier curve to return to drug use, it's a better, what you just said is better than what the FDA approved with lofexidine. I'm optimistic, but it's not a cure. It is not a cure.
Thanks, Dr. Mash.
Did I say that enough? I wish it was. It's not.
Azim, did you have a question?
Yeah, so Dr. Mesh, I may have not completely caught this, but I wanted to just understand again how you plan to use the noribogaine. Is it like, let's develop Ibogaine, make sure it works, and then we can try to use noribogaine as well for the same purpose of sort of that initial detox phase, or is it going to be more of a maintenance treatment?
It's a really great question, and thank you for asking it, as obviously I wasn't clear. This goes back to the oniric, all right? So originally, we reasoned that, okay, Ibogaine is a Schedule I drug. You're not going to get to work with it anyway. You don't have any IP. Now you have noribogaine. You have IP. It's not an oniric. You could do an office-based detox. Doctors could do it in the office, could hook them up to a cardiac monitor, give them some noribogaine, bring them back, long half-life. You could do it. You could imagine a protocol, and we did. We've walked away from that. We've come back around, and it's interesting, when we first started out with this, and we had the counselors down in St. Kitts and the therapists and the psychiatrists, and I would argue, as a pharmacologist, we don't need the visions. We don't need that. And we got a molecule, it's got targets, we know what the affinity, we can do this, we can make a pharmaceutical, and off to the races. So I argued that. Visions, we don't need that. Today, I sit here, we absolutely need that. Completely, I've come full cycle around about this. And I think that, people are out there microdosing Ibogaine, right? They're doing it. They're taking low doses of Ibogaine. What's in their blood when they take low doses of Ibogaine? Answer, noribogaine. And by the way, when people in France went to those little pharmacies and bought Lamborine, they were walking around with only noribogaine in their blood, you see. So yes, it should be a maintenance therapy. Low dose,
Okay.
Low dose, well below any hERG signal. Dr. Rudnick says, you've got the targets, you've got the affinities there, it's potent. He said, what is the potency? The potency is going to be better, and you can come in lower on the concentration, get below the hERG signal, and have a maintenance therapy. Yes.
Got it. And maybe you can clarify that sort of change in perspective you had about the visions. Why did you change your mindset on that?
Why did I? Because when I said this in the clinic, my therapist looked at me and said, no way. Okay, no way. This is part of the therapeutic process. You're taking people who have abused drugs for a very long time, who have been locked in an intractable cycle of use and a couple months of clean time, back out using again, on Suboxone, off Suboxone, back on Suboxone. It's not going to work. And I think that if we think about why people become addicted, why us on this call don't become addicted and why others do become addicted. We know that there are many issues here from the field of psychiatry. You know them better than me. You've got the comorbidity. We already said this with depression and anxiety. People are self-medicating. They're also self-medicating inner wounds, the traumas. And Ibogaine, the new kind of renaissance that we're seeing with psilocybin, MDMA for PTSD, et cetera, that we can use these, that qualified physicians can begin to use this class of molecules as adjuvants to the behavioral intervention. And with that, you've got a winner. We need to have people engage to rewrite their own mythology, to take control of their life, and to feel that they can succeed. What is going to be different this time? I've tried to detox 5, 6, 7 times. My family has run up, huge bills. They've taken money off their mortgage. They've taken second mortgages to put me in, to save my life and put me in treatment. Maybe Ibogaine is that oniric effect, giving people the insight into their self-destructive behaviors. The patients told us, they said, Dr. Mash, this is like a 4th step, completing a moral inventory. So they were able to go back into their family and say, I'm sorry, and to begin to work a program and to stick to it. That's our hope. That is our hope.
Yeah, well, it certainly stands to reason, and I think it goes with a lot of people's thinking about can we make a serotonin 2A agonist that doesn't produce these effects, and will it be as effective, and those types of discussions. So it's it's interesting to hear that you guys have sort of come down on the side of the importance of that experience for folks to change. Are you all collecting data through any collaborators about this sort of microdosing approach and its ability to keep people abstinent? And
we hear it. I want people to be able to take Ibogaine in a safe setting. I've said this a lot. I say it all the time. I say it, I'm like a broken record with this, but I want patients to not have to go into these underground, unsafe settings. I just heard last week about a woman in Mexico who's had several deaths with Ibogaine, who will now only give low-dose Ibogaine to patients. And apparently she's getting people off of nicotine with low doses of Ibogaine. I think they can just go do these things, So I think the low dose experiment was done in France. Okay. And I think that low dose Ibogaine is Ibogaine, and we know that the pharmacokinetics is there.
When do you think that it'll be after you sort of have demonstrated efficacy of Ibogaine initially that you'll plan to start those studies?
Good, very good questions. You want to come work for me? Those are excellent questions. Yes, that makes perfect sense from a collaborative and from a best practice perspective. We're not going to jump back in full boat until we can get these results in humans with the Ibogaine and safety, patient safety, learning a little bit more about this, working collaboratively with the regulators to come up with the best protocol. And then, of course, we are continuing to push Ibogaine forward as well. But, it's funny, I spoke to Charles Krajinskas. I know some people on this call may know who he is. And I called him and my former medical safety officer from the FDA, who first approved us to do Ibogaine, Dr. Dan Spiker. And they're both in the same company now, working as, they have a company and they're helping people who want to develop drugs. And he said, Deborah, he goes, what is wrong with you? You cannot develop 2 molecules at the same time. So I paid attention to Charlie, and we're staggering them.
Fair enough. So I'm gonna ask a question from the chat here. Shinwan says, could you expand on the elicitation narratives from the St. Kitts study? And he wants to know, how explicit was it? For example, did you ask the patients to focus on specific things? And then he also expands and asks if if there's any correlation between those self-reports and individuals' recovery.
Yeah, it's a great question. Thank you for that question too. Yeah, I'd be honest with you, when we were doing that, I mean, we didn't know what we were doing because, nobody had anything like that. And we needed to we wanted some way to start to operationally define, the information, and without a good idea of how to do it. So that's why we use the anthropologic approach. And yes, we had a set. The patients were asked the same questions, and then they just were, and this was done under video and recorded. So the patient was asked the question, and then they started to answer the question. That was transcribed, and then content coding was done by 2 psychologists. So that's how it was done. And I show a little bit of that data in the 2018 paper. We have a lot more of that data. Did we follow up on it? No. Are we going to use the elicitation narrative in the clinical trial? Yes. So if you have ideas or you want to collaborate, get back in touch with me. Email me. Very good question. Because I think we can get that, together with the readiness for change data. And I won't go into more details about the protocol, but it will be used in the upcoming study.
I think that's great that you were collecting the qualitative data as well. Happy to hear that. And while we're sort of talking about the one-iric effects, as you were referring to them, I was kind of curious to ask about the use of that term. I think most of us in this is the psychedelic science group. I just thought it was kind of interesting that you're preferring that term over and maybe avoiding the term psychedelic, or just curious to ask about
Could be.
if it's more of a specific decision, and also if there potentially are phenomenologic differences.
There's great questions. These are so great. So obviously there was a little part of me that was trying to originally distance, from everything else out there, especially when we first went up in front of the FDA. But what does that term mean? It means dreamlike, right? And the patients told us it's a waking dream state. I don't know, because I've never taken Ibogaine, and none of the people that worked for me were allowed to take Ibogaine either. So I was kind of a little strict, let's put it that way, about this. And I'm still strict about it. But anyway, so I don't know. But the thing that they described over and over again is that they would say, well, some of them would hear a high frequency pitch sound, and then they said it would, the screens would come down. It was like a life review, watching a movie. And we heard that over and over and over again. And I think that was described by many others in the literature, and Latzoff described it, everybody described it, but you kept hearing it over and over again. So many of our patients, of course, had taken lots of things. So they were not hallucinogen naive, let's put it that way. So was it LSD-like? Was it psilocybin-like? Was it MDMA-like? What was it like? And they all said it was quite different. The second phase of it, when noribogaine is really high in the blood, I think might be, I don't know this to be true, but my gut impression from listening to them. So the visions, when the Ibogaine comes on, the visions come on really hard, and then depending on metabolizer status, fast, slow, or intermediate, they'll stay in. And then when the curves cross over, the visions abruptly stop, and then they go into a deep phase of cognitive introspection. And they like it. It's kind of like, it must be, I think it's what Gary was talking about, lots of serotonin. Because that sounds like that heart chakra opening thing that we've heard with other molecules. And they reported that too. So, and then that would wear off. But again, they were thinking, there was a lot of cognition going on there. And we kept them quiet. We keep them in bed. We didn't talk to them. So I think of Ibogaine as in a separate box. And I had this conversation, I think we talked about this, you and I, on the phone. I had this conversation with Sasha Shulgin, the late Sasha Shulgin, and he explained to me, and he said that one box was, psilocybin, tryptamines, one was LSD, one was the hallucinogenic amphetamines, and Ibogaine sits alone. I think it's different. I definitely think it's very different. But having never taken it, I can't say that I know that to be true.
Does it have serotonin 2A agonism in it?
Ibogaine, according to the literature, there is a little 2A on the Ibogaine, yes. Noribogaine, no. Noribogaine hits no serotonin receptors.
Yeah, no, it certainly seems from reports that it is different, and I was intrigued by the piece of data that was in your paper and that you shared, that such a low percentage, only 16% of the folks said that they would actually be willing to do it, even though that they reported efficacy or that it seemed helpful for them. And in the psilocybin literature, we know that it's not considered a drug of abuse and doesn't show that kind of habitual use. But my hunch is that, at least from the research I've done with depressed patients, that the rates of wanting second doses is probably much higher in at least in certain indications, and with psilocybin potentially than.
Well, and I think, we need to explore this. Of course, if this is a neurochemical reset and there's an underlying, the neuroadaptation, depending on whether we're not remodeling the entire brain, we're resetting a homeostasis. And it's giving you a window. Ibogaine is an addiction interrupter. It's giving you a window of opportunity to get in there and do the other things, like deal with the underlying neuropsychiatric disorders that got you to self-medicate with opiates in the first place. Okay, that would be a good place to start. The behavioral counseling, the dealing with the traumas or whatever it is that's in the wheelhouse that's making you develop a coping strategy that includes opiates. So I think, people may need multiple doses. I can tell you that I don't know, and I've known people who've taken multiple doses of Ibogaine at different times. I know people who've relapsed and taken Ibogaine again years later. I think that's okay. Because as we all know, some of us, addiction is a chronic relapsing disorder. And it's a chronic relapsing disorder.
Thanks for sharing that. So I'll ask the audience if there's any last questions, moving towards wrapping up. Since there hasn't been that many, I'm glad I've been able to ask all of my questions. I'll ask.
Jordan, I have another question if we have Dr. Marsh for a little bit.
Yeah, we have till the top of the hour.
Yep. Dr. Marsh, do you have a sense if any of your patients who are in that Noribogaine treatment, any of them who had an early relapse to heroin, if there's any self-reported protection or lower amount of
These are great
high that was gotten from it, just as we think about the sort of dangers of not using a substitution approach?
Yes, I think that's a great question. And obviously the drug interaction studies would need to be done, clearly. From a mu standpoint, it's really not much of an agonist. So I'm not too worried about that. But again, those kinds of studies, that has to be considered.
Mm-hmm. So no animal literature that shows some sort of protective effect or something?
No. There's some Noribogaine at really high doses will, in combination with methadone, I think we have that data if I'm not mistaken, and I think we've done that, could lead to, increased lethality. Now, is there an interaction with benzodiazepines? I don't know. I don't think that's been well worked out. But as with Suboxone, or if you use drugs, if you take benzos and you're on Suboxone, you can get in trouble, right?
Mm-hmm. Mm-hmm.
So people do OD on drugs.
Yeah, I guess the sort of more of the flavor of question that I was trying to ask is, if someone is on Suboxone or on methadone and they relapse, there's a relative amount of protection that's afforded to them, as opposed to, let's say they got a naltrexone injection, or they got a naltrexone injection and they miss their injection, or their motivation decreases, they're a week out, all of a sudden they don't have that sort of protection that they might have on
they go out and use
Suboxone.
They can have an accidental overdose.
Right.
And that's and we heard a lot. We know about this. It does happen. I can tell you that most people who are career, injectors know how to dose. They know how to dose. Okay. And why did we see that uptick in the deaths? Fentanyl, right? The fentanyl analogs. Fentanyl analogs were in so many of the opioid-related deaths in South Florida, various ones.
Mm-hmm.
So
Roger's asking a question here that might dovetail on this. He's asking if the protocol changes in cases of heroin mixed with fentanyl versus fentanyl alone.
It's a great question. It's an extremely important question because fentanyl has a long half-life and it has an active metabolite. So yes, there would have to absolutely be a washout with fentanyl. Absolutely. And we will be looking for fentanyl.
And we have a question from Dana that's a little bit of a
especially in the early
separate
safety, especially, we need to control for that very, very carefully in the early safety work we're doing as well. It's a very important thing. Yeah, fentanyl, I hate it. Hate it.
Thanks, Dr. Martian. And we have a question from Dana that says, new treatments for drug dependency are not on the platform of either party. What step will you take to change that?
Oh, good Lord.
So not only drug development you have to sort of deal with, but a little bit of lobbying here too. So what are your plans?
No, that's, and I appreciate the activism. All right. I really appreciate the comment on this. I was an elected official in my municipal government and I was term limited out. So I've done that. And I've been very involved in politics. My ex-husband was chairman of one of the local parties. You can guess which one. I'm not going to name it. So we were very, very, very involved in politics. And I spent a lot of my life being an activist and being involved in politics and working the polls and helping get people to the polls. And by the way, everybody get people to the polls because we got to make sure people vote again this time around. So it's very important. Have I spoken to government? I have. Have I, do I think that there's a place for this? I think the most important thing right now is to, I've been laying low, really laying low and not, bringing too much out there to the, to politicians, let's put it that way. I think it's much more important that we get in the clinic, that we see, that we do the pivotal studies, quickly get in the clinic, demonstrate the safety, and go for the efficacy endpoints. Bring in the stakeholders, bring in the colleagues and the peers, the people who are involved in addiction treatment in our country and ex-US, and position the drug in that way. And we'll quickly know whether this is a go, we'll have go, no go decision very quickly. So I don't want to overpromise, and I don't want to overstate it. One thing about being a good scientist is, you have to step back from your data and let the process go forward. This needs to be studied in a controlled, blinded, placebo-controlled trial.
Absolutely. And we can all appreciate that we, just given the sort of psychedelics revolution, if you will, we live in a much different world than we did for development of these compounds than in the 90s. And even with potentially the 18MC story, though, it seems like you're alluding to more of a efficacy question there than a regulation question. But I'd be curious what lessons there you have to share in terms of things that you've learned in terms of dealing with NIDA and the FDA for the development of these compounds that you're sort of incorporating now.
Thank you for asking that. I'll comment on that. FDA was amazingly collaborative. They always have. They always have been. I've been up in front of the FDA 4 times, twice for Ibogaine, twice for Noribogaine. Very collaborative, extremely helpful. And don't forget, they approved Ibogaine to be studied. The reason we didn't go forward was because NIDA didn't think it was a good bet. And at the same time that, NIDA shifted gears on it, because NIDA looked at it, they spent money on it. They did preclinical studies to support a clinical trial application to the FDA. They participated in my meetings with the FDA. I had NIDA sitting right alongside of me as an academic investigator. So, the NIH, we want the NIH to fund all the best science that we have in the United States. And drug development is very, very, very expensive. The NIH does, collaborative trials in oncology and whatnot, but there wasn't the money in the National Institute on Drug Abuse to fund a lot of studies. They had to be very certain about this. At the same time, there were groups. I think Dr. Leshner signed the CRADA with Reckitt on moving buprenorphine forward for office-based practice. So this was all going on all at the same time. So, we can't expect the NIH to take a drug to approval. I mean, every now and again, you hear wonderful stories about how that works. And brexanolone, the drug for postpartum depression that I mentioned, that came from, I believe that came from NIMH and then was advanced. And so that's really amazing. And I've been a NIDA-funded investigator for a very long time, and I've been NIH-funded, and I'm still NIH-funded today. I'm still working with collaborators on epigenetic studies and postmortem human brain and providing the brain tissues for that. So I'm still participating in that and enjoying it. But we can't expect, again, the NIH has limited funds. They need more money, and we should be politicking for that, because American science is always a great bet. So let's do that in the context of the next election. Let's make sure the NIH dollars continue to flow. And the FDA, I'm optimistic. The FDA, I think they want to help. I think they always, the people, the men and women that work at the FDA, they want to help. They want to advance drugs. Is it easy to get drugs through the FDA? Hell no. Oh, hell no. Oh, hell no. But we are the market. I mean, the big market share for drug abuse is the United States. It's not Canada. It's not Europe. It's not Australia. It's right here. Americans love their drugs. And we need more Americans back in their families and in the workplace, contributing as taxpayers.
Well said.
Okay, I think we should just go home.
Yeah, I think that's a really great place to leave it. Dr. Mash, I'll read you the comment from Kate Becker saying, which I think represents everyone's sentiment. Thank you for the presentation and for being an amazing pioneer in this work. Really want to thank you for your time being with us, and really hope that we'll be able to have you back in hopefully not too many years when you have your data from these upcoming trials and you have the good news to let us know that Ibogaine's rolling out as a breakthrough treatment for opioid use disorder. So best of luck with all those endeavors. And again, just thank you very much for spending your time with us. It was so informative and a really wonderful presentation. We had close to 70 people on the talk at one point, so really nice turnout.
Thank you very much. Thank you for the opportunity, and I really enjoyed the questions. It was good to see some old friends, and everyone stay well, stay healthy.
Take care, everyone, and we will be back next week with another webinar with Dr. Brian Anderson from UCSF, who's going to talk to us about his work with psilocybin therapy amongst AIDS survivors using group therapy. Should be another really interesting talk. We'll be sending out some information about that. And until then, thanks again, Dr. Mash, and hope everyone out there is staying safe and healthy. So take care, and signing off for now.
Signing off.